Hi, everyone. Today we're going to be presenting our research from this spring. My name is Jake, and with me I have,
I'm Maddie.
And I'm Hannah.
and we're going to be presenting protein structure analysis using reverse phase HPLC. In the biochemistry curriculum, the understanding of structure-function relationships of proteins is pretty vital. In order for students to understand these relationships, it is essential for them to understand protein structure and their different conformations. With this in mind, we are developing an undergraduate biochemistry laboratory experiment to enhance students' comprehension of protein conformations, redox reactions, and high-performance liquid chromatography. Through the use of reverse phase HPLC with a carbon 18 column, different conformations of lysozyme were analyzed this spring. And with a lysozyme protein, when it's completely unfolded, the native conformations have exposed polar surfaces, giving a lower affinity for the column and the shortest retention times. The proteins are partially unfolded from different influences. They expose parts of their interior hydrophobic residues, allowing them to have longer retention times. When the protein is fully reduced and is completely unfolded, lysozyme has the longest retention times in the column. The visualization and contrasting elution times or retention times can help to further reinforce the understanding of protein conformation and its influences. Our objectives in this was to develop a method in which reverse phase HPLC was analyzed and the retention times of lysozyme was used to illustrate the different structures of lysozyme. So what is HPLC? High-performance liquid chromatography is an analytical technique to separate and identify components in a mixture. Separation in reverse phase HPLC depends on the hydrophobic binding of the solute molecule from the mobile phase and the ligands attached in the stationary phase. The ligands covalently bound to the stationary phase provide a hydrophobic surface. The mobile phase passing over the hydrophobic stationary phase is a polar solvent containing the solutes, or in our case, the protein. We're using a C18 ligand, which is a hydrocarbon chain that is 18 carbons long and is attracted to nonpolar or hydrophobic molecules, or to attract nonpolar or hydrophobic molecules. So in this diagram, you can see an overview of the HPLC system. The reservoir holds a mobile phase. In our mobile phase, we are using a blend of acetonitrile and deionized water. Acetonitrile is used to maintain the polarity at a low enough level for the protein to dissolve, but high enough to facilitate binding to the stationary matrix or column. From the mobile phase, a high-pressured pump generates a specified flow rate of the mobile phase. For our method, it was 1 milliliter per minute. The injector is used to inject the protein sample into the flowing mobile phase stream that carries a sample to the HPLC column. The detector is needed to see the separated compound bands as they elute from the column. The detector is wired to a computer that records the electrical signal needed to generate a chromatogram on this display. For our results, we examined the UV detection at a wavelength of 280 nanometers. After exiting the detector, the mobile phase is sent to a waste beaker or container. In this picture, you can see our setup, a little less simplified, but all of the same things exist. So what are we analyzing? Generally said, we monitored how long in minutes the sample remained in the column before eluting. And then the chromatograms for this were evaluated on the computer software at a wavelength of 280 nanometers.
So protein structure is a very important part of our project as a whole. The building blocks of proteins are called amino acids, and they are formed in a chain of polypeptides. There are 20 types of amino acids, and these acids determine the protein's shape and function. This diagram is showing us the the structure of an amino acid broken up into the amino group and a carboxyl group with a side chain. Proteins go through different structures as they are forming themselves. The first is called the primary structure, where amino acids are linked together by a peptide bond in a chain. The secondary structure involves interactions with hydrogen bonds between the carbonyl O of the amino acid and the amino H of another. The most common types are the alpha helix and the beta pleated sheets. The third structure is called the tertiary structure, and it is known as native conformation. It is a 3D shape that has various interactions between amino acids, R groups, and other molecules. All of these bonds are non-covalent and they are hydrophobic interactions. Disulfide bonds are part of this third structure. They are covalent bonds between sulfur residue cysteine R groups, and they are very strong. These disulfide bonds are where the reducing agent breaks apart the bonds in our experiment. Throughout our experiment, we observed 3 different stages of proteins: native, denatured, and reduced. The native protein is in its 3rd stage of formation, and it's also a monomer. All of its bonds, including peptide, hydrogen, and disulfide bonds, are intact. Inside of the structure are the hydrophobic and nonpolar regions. These proteins should have a short retention time and not have a high affinity for the column. Denatured proteins is the disruption of this 3rd structure and the secondary structure to cause the proteins to go back to its original primary structure of the amino acid chain. The amino acid sequence is still intact, and so are the disulfide bonds. The cause of this denaturing includes heat shock, extreme pH, and urea. This denaturing exposes the hydrophobic regions of the protein, which gives it a longer retention time and a greater affinity for the column. The final protein we looked at is the reduced protein that disrupts the disulfide bonds, but the primary structure remains intact. The cause of this is DDT and 2-mercaptolithiol. This reduces and fully unfolds the protein so there is full exposure of the inside regions, and it has the greatest affinity for the column and the greatest retention time. DTT is the reducing agent we use to get the protein to its reduced form. This is the structural pathway DTT takes to reduce protein. The next slide gives an overall view of the 3 different types of proteins: native, denatured, and reduced. You can see, as I said before, the different qualities of each of these proteins and how they interact with the column. The protein that was used to do this experiment was lysozyme from chicken egg white. It is a globular enzyme, and it is small and stable. It attacks the cell walls of bacteria to break down carbohydrate chains and destroy the cell. It also has internal disulfide bonds, and it is present to protect the nutrients in the developing egg of a chick. In the final slide, you will see the disulfide bonds of the lysozyme structure and how they separate to expose the inside of a protein to have the strongest affinity for the column in its reduced state.
So for our procedure, we ran all 3 of the proteins at different times to see their retention time. First, we ran the native lysozyme. The native lysozyme was put in a citrate buffer with a pH of 5.2, which is the optimal pH of lysozyme. The solution with lysozyme had a 1 milligram per milliliter protein concentration, and our injection volume was 20 microliters. Our 2 mobile phases were water with 0.1% TFA and acetonitrile with 0.1% TFA. For denatured, the same buffers, solvents, and method was used. To get the protein to denature, we used 1 milliliter of 10 mol urea with 1 milliliter of 2 milligrams per milliliter lysozyme in a vial for the final concentrations of 5 mol urea and 1 milligram per milliliter lysozyme. For our reduced form, we still had the same buffers, solvents, and method, but we combined 1 milliliter of solution, which was 10 mol urea and 0.2 mol DTT. We combined that solution with 1 milliliter of 2 milligrams per milliliter lysozyme in a vial for the final concentrations of 5 mol urea, 0.1 mol DTT, and 1 milligram per milliliter lysozyme. So for our method, the variation in concentration was of acetonitrile. For the first 5 minutes, there was 38% acetonitrile, and the purpose of this was for equilibration and to allow the elution of buffer and other erroneous substances. For the next 18 minutes, we would ramp from 40% to 60% to increase the acetonitrile concentration, which displaces the protein from the column, causing elution. For the following 5 minutes, there would be 100% concentration to remove anything else that could possibly be left behind in the column. And the last 5 minutes it would be brought back down to 38% to re-equilibrate. So our experimental results that we got this spring. So we ran 4 runs. First, we ran a blank, which was 1 milliliter of citrate buffer with 1 milliliter of 8 mol urea plus 0.2 mol DTT solution in order to make sure that the peaks we were getting were indeed from the protein. For native, it was, as I said before, 1 milligram per milliliter lysozyme and citrate buffer. For our denatured protein, we combined 0.5 milliliters of 2 milligram per milliliter lysozyme and buffer with 0.5 milliliters of 10 mol urea. And this was incubated at room temperature for 35 minutes before injection. For the reduced form, we combined 0.5 milliliters of 2 milligram per milliliter lysozyme and buffer with 0.5 milliliters of 10 mol urea and 0.2 mol DTT in solution. This sample was heated at 55 degrees Celsius for about 72 minutes before injection.
Just a correction, in the blank, it was 10 molar urea.
Thanks, Jake. As you can see in our blank, these peaks are all from our solutions and buffers. For our native protein, it had a retention time of about 10.5 minutes, and you can see that in the graphing. Our denatured protein had a retention time of about 10.8 minutes, and our reduced protein had a retention time of about 13 minutes. Some conclusions, our denatured form of lysozyme was not reproducible in the time we had this semester. If we had had time to continue, the concentrations on urea could be increased along with temperature influences for the denatured phase to try to get our desired results, and the method could also be further adjusted if need be. What else we learned? We learned that the denatured sample must be given the appropriate amount of time to work and most likely needs heat to fully react. Reduced samples need to be heated for time constraints, and the concentrations of urea may need to be increased to provide desired denaturing effect and better separation.
Okay, so moving forward, this was my last semester with research, but I think this experiment and research is going to be moving far past me and with other students, great students just like this. So to move forward in this research, I think adjusting the method and procedure is needed to help more illustrate the separation of denatured and native forms of lysozyme, as well as other proteins like alpha-chymotrypsin or just trypsin inhibitor. And then also to write up and develop a laboratory experiment protocol would be very helpful so that other students at other colleges and universities could do this procedure to help illustrate the different structures of proteins in biochemistry. And we'd like to acknowledge a lot of help from other people in this research. First of all, Dr. Charlotte Boy, Jane Schubert, she was a great help. And then past students like Emily Welsh and Lan Nguyen, and the Nazareth College Departments of Chemistry and Biochemistry, and all other students that previously worked on this project. Thank you.